Hello, good morning, everybody. Can you hear me? Hopefully you are able to hear my voice. I am your host of the event today. My name is Jeffrey Poon, the Regional Product Manager for Biosafety Carbonate, CO2 incubators, and biological shakers for Asia. I am based in Hong Kong. 2 weeks ago, we had the first live webinars about how we can transition research to GMP production, particularly if startup companies are on their learning curve of the whole migration process. So last time we had a first talk on the workflow of gene and cell therapy and a short overview of what we should look for in a CO2 incubator. Today, we are going to cover a different topic, how a biosafety carbonate can meet the GMP requirements. So we are very pleased to have Dr. David Phillips to give us a live webinar from his home, which is 12 hours behind us. Just a few house rules before we start. You will be muted during the event. However, there is a Q&A box on the right-hand bottom corner of your screen. You can type in your questions, but I would suggest you to send your questions privately to preserve personal data privacy. So we will have about 10 to 15 minutes after the presentation for Q&A sections, so that the whole event will last for approximately 1 hour. Now today, the speaker is Dr. David Phillips, our Senior Global Product Technology Specialist for BSC and clean benches. Dr. Phillips has been involved with clean rooms and biosafety carbonates for almost 40 years. So he worked with nationwide contamination control service company in a variety of roles, including until 2007 when he joined the Thermo Scientific's BSC product team. So he is now one of the NSF Joint Committee members for the NSF NC49. He is the first NSF accredited BSC field certifier since 1993. So he has a doctorate degree in management from the University of Maryland. His key area of interest and even his favorite pastime are in improving BSC sustainability and access to field certification globally. He is very crazy with BSC. He could even spend his annual leave traveling to Azerbaijan and certify a carbonate. So without further ado, I will pass the time to Dr. Phillips.
Well, thank you, Jeffrey. I appreciate the introduction, and as you can tell, I really like this subject. Welcome to this presentation on GMP and Class 2 biosafety cabinets. I find it very interesting, and hopefully you will too. And I'm especially looking forward to your comments and questions at the end, because it's always fun to learn from others. Let's see. So GMP, as we see in this definition from the International Society for Pharmaceutical Engineering, GMP, or good manufacturing practice, is a system for consistent and controlled production of drugs according to quality standards that minimizes risks that cannot be eliminated in testing the final product. GMP are the processes and procedures required to produce drugs. Regulatory bodies like the U.S. Food and Drug Administration and others inspect manufacturing producing drugs for use in their countries to assure they are following GMP or CGMP. CGMP is current good manufacturing practice. GMP is heavy on procedures, processes, documentation, and validation. GMP is focused on the production of safe and effective drugs. It does not directly concern itself with personal or environmental safety. Facilities also must assure safety, but these are different regulations and are addressed in other ways. Let's see. The Class 2 Biological Safety Cabinet, or BSC, was developed to enable the safe work with potentially biohazardous agents like tuberculosis, the coronavirus, and other infectious viruses and bacteria. It was not born in the GMP world. The Class 2 BSC draws air in at the front opening, as you see in the diagram, those green arrows at the front, and that inward flow of air prevents any airborne hazards inside the work area from escaping the cabinet. But that inward flowing air from the room may contain contaminants, so all of it is drawn into the front intake grill. From there, the room air is pulled underneath the work surface, up inside the rear wall into the motor plenum above the work area. From there, some of the air is pushed through an exhaust HEPA filter, and the filtered, particle-free air is returned to the room. Those are the blue arrows coming out of the top of the cabinet. The remaining air, about 60 to 70%, is pushed through the downflow filter into the work area, becoming this particle-free, ISO class 5 or better, smooth, unidirectional airflow. Those are the blue arrows you see within the cabinet. A Class 2 BSC is a partially contained area, not totally contained like a barrier isolator or glove box, not uncontained like an area within a clean room, but partially contained. By restricting the access to this to the work area with this 20 to 25 centimeter tall front opening, we gain a validated air barrier that contains hazardous materials, a cleaner work zone with lower air velocities, and a greater capacity to monitor that processing environment. Its initial development as a safety device still influences many of the practices and uses of these devices, even as we have sought to exploit the particle-free and contained work area within the cabinet for GMP. For me, the two greatest examples of this safety heritage are the types of Class 2 BSC and the field certification. There are currently 5 types of Class 2 BSC under the NSF ANSI 49 standard. Where did they come from? The Class 2 biosafety cabinet was developed to enable the safe work with hazardous viruses and bacteria, which are particles and are easily captured by the filters. This was our first type of Class 2 BSC shown here. But we wanted to safely work with volatile toxic chemicals, but gases pass through the filters. So within the NSF standard, we developed a type of Class 2 that was connected to an external exhaust system, kind of like a fume hood. We called the first one a Type A and the one for volatile chemicals the Type B. The Type B was safer for work with volatile toxic chemicals. Unfortunately, some began to think that safer for a specific issue was the same as better. It is not true for BSCs, just like it's not true that just because rubber boots are good in the rain, that they're better for everyday use. As time passed, people began noticing that even pharmacists using these Type B cabinets when preparing cytotoxic drugs were somehow being exposed to these hazardous drugs. Some thought the droplets of the cytotoxic drugs were getting captured by the filters, including the downflow filter, which was true, but then would off-gas and turn into a hazardous gas, re-enter the BSC work area, and somehow penetrate the skin of the pharmacist's hands or arms that are in the BSC work area, or somehow escape out the front opening and get into the pharmacist. This is unlikely and has never been demonstrated, but the idea was popular, so an even safer exhausted cabinet was developed that had no internal filtered recirculation. The first BSC for chemicals became the B1, and this even safer version became the B2. Later, we modified the Type A to make it possible to connect to external exhaust and thereby allow work with volatile toxic chemicals. And later still, there were so many problems with the B2, we came up with a less demanding version that still had some non-recirculation as a C1. But see how this process is driven by safety, personal and environmental protection, and not product protection. There's nothing wrong with that, but in the GMP world, we're more interested in protecting the product, and we need to remember that when it comes to BSC development, safety is in the driver's seat. Field certification is another example of how safety is the focus of biosafety cabinets. Within safety devices, It's not uncommon for the maintenance scheme to be regular inspection where any found deficiencies are corrected. When I used to live in Pennsylvania, that's how they handled automobiles. We needed to get inspections every couple years. They would tell us if our tires needed to be replaced or brakes serviced and so on. The theory behind this approach is as long as you correct problems before too much time has passed, overall you will have acceptable levels of compliance. A problem for us in GMP application is that it's not acceptable to the agencies regulating the safe and effective production of drugs. It's not helpful to tell the FDA that we monitor our process and correct any problems after only allowing a few contaminated doses to be produced and distributed. If the heritage and development of Class II biosafety cabinets does not align with the GMP arena, why are they used? Well, first, the work area is beautiful. Particle-free to ISO class 3 or 4 with an easy ISO class 5. The airflow pattern is unidirectional and defined. The work surfaces within the BSC are usually stainless steel and glass, and some models have even a higher grade of stainless steel, type 316. The partial enclosure builds in a nice degree of control. On top of that, biosafety cabinets address a growing need. Year after year, we are developing more and better drugs, more powerful, more concentrated, and more needed than ever before. The potential negative effects to the people working in our manufacturing facilities with these potent compounds just keeps increasing. And well, what do you know? The Class II BSC is designed to contain hazardous materials. The device starts to be pretty appealing, but we need to figure out how to make it a good citizen within the GMP working environment. One of the characteristics of the GMP environment is that it is very engaged with the process. We do not take the view that we can make a drug however we want and then test it to see if it's okay and then send out the okay ones. Using the US CFR as an example, Section 501A 2B states, a drug shall be deemed adulterated if the methods, facilities, or controls used are not in compliance with cGMP. In other words, if the process is flawed, the drug will be deemed adulterated. Drug manufacturing is not a black box where the ingredients go in one end and the drug comes out the other, and as long as the drug is correct and uncontaminated, no one looks in the box. The GMP approach makes everything that goes on in the box of interest. The ends do not justify the means. The means must be provably correct, and the ends must be provably correct. The US FDA published a guidance for industry with general principles and practices on process validation. In that document, the FDA makes it clear that in their view, the specifications for written procedures and process control require process validation. So within that black box of manufacturing, the processes used must be thoughtfully designed and validated to produce what we require, including that the equipment used is of the appropriate design and the written program describing its use, maintenance, inspection, and calibration is designed to assure proper performance. This guidance breaks process validation into 3 stages. Stage 1 is process design, which has 2 parts. The first part, building and capturing process knowledge and understanding, allows us to use existing information and technology as we begin to put the process together. We can imagine that it is here where BSCs made the leap from biohazard control to use in pharmaceutical production. The process designers could note the BSC sample chamber with the ISO class 5 air cleanliness, coupled with the proven air barrier at the front opening, and then bring those attributes into their processes. One of the benefits in using a Class II biosafety cabinet in this application is the existing validation scheme. There are recognized Class II biosafety cabinet standards, NSF ANSI 49 in North America, EN 12469 in Europe, and YY 0569 in China that include specific requirements for design, construction, and manufacturing, coupled with type testing validation of performance. Thermo Fisher Scientific supports the use of our Class II BSCs in the cGMP environment with augmented documentation packages under our CTS or cell therapy systems containing 1, our certificate as to the materials of construction, our CE declarations of conformity, CB test certification of conformity to IEC 61010-1, including eligibility for the CSA mark, our TÜV Nord certification for compliance with EN 12469, our commitment to REACH compliance, TÜV SÜD certification of our cleanroom compatibility up to ISO Class 5 per VDI 2083 Part 9.1, as well as TÜV SÜD certification of our quality management system of ISO 9001. Also in stage 1, we need to determine our strategy for controlling the process. At this point, we're going to step away from the process validation document. We'll come back later, but we want to look at what are the likely requirements of the process for our Class II biosafety cabinets. There are two general approaches to producing uncontaminated drugs. The traditional approach is terminal sterilization, where the product is filled and sealed under clean conditions and then sterilized by heat or irradiation. But some drugs can be affected by the heat or irradiation and so are prepared under aseptic conditions. Aseptic processing brings the clean components together and assures the cleanliness of the final product by doing so under aseptic conditions. It is production using aseptic processing where Class II BSCs are typically used. There are two documents with some of the most common guidance on the environments for aseptic processing. The US FDA's Guidance for Industry, Sterile Drug Products Produced by Aseptic Processing, Current Good Manufacturing Practice, which first hit the street in 1987 and it was then revised to its current form in 2004. In Europe, we have Annex 1, Manufacture of Sterile Medicinal Products, from Volume 4 of the EU Guidelines to GMP Medicinal Products for Human and Veterinary Use. The first version of this was dated 2003, and this latest version is effective as of 2009. These are very similar. For simplicity's sake, we will spend most of our time with Annex 1. One of the first statements in Annex 1 lets us know what we're in for. Notice it is primarily talking about clean areas. The annex is speaking to clean rooms more than to clean air workstations and certainly not biosafety cabinets. It does include cabinets and isolators and such, but we need to remember our cabinets are not the primary focus. We can make it work. For example, when it talks about how clean areas should have entry through airlocks, we can kind of see the air barrier at the BSC front access opening as performing that same function. Other phrases are easier. For instance, a BSC definitely maintains an appropriate cleanliness and is supplied with air passed through filters. But we need to translate from pharmaceutical clean rooms to biosafety cabinets. Another idea we need to get used to is occupancy class. What we care about is the cleanliness of the environment while we are manufacturing or processing the drug, or as termed here, in operation. But one of the key ideas in cleanroom operation is that the level of cleanliness varies with the activity in the room. I saw a chart once that showed the levels of airborne particles in a cleanroom over the course of a day. It looked something like this, where you would see very low levels overnight, a spike as people came in and settled down to work, then another spike as they got up for lunch. When they return, it settles back down to an operational level, not as low as overnight, but lower than when everyone is coming and going. Another spike at the end of the day, and a huge spike when they come in for cleaning. After the cleaning, when no one is in the room overnight, you see the lowest levels and it will start all over again the next day. In the cleanroom world, we're very aware of the occupancy state, and sometimes in order to get the operational environment we want, we will specify a cleaner environment for at rest. We see the groundwork for that here, where they state there will be a specified cleanliness level for the at rest condition, and they go on to define exactly what they mean by at rest. What is operational and at rest in a Class II biosafety cabinet? Well, operational would be while someone's actually working in the cabinet, and at rest would be when the cabinet is on but no one's hands are reaching in. This is tricky for us. We hardly ever do any testing or measurements while people are actually working in the cabinet. Annex 1 defines 4 grades of clean rooms for the manufacture of sterile medicinal products. Grade A is the cleanest, and it is for the moment the drug is most vulnerable to contamination. An example would be the filling zone when they're putting the drug into a container. If anything gets in there, it will be in the drug when it's administered to the person. Same thing for open ampules and vials and so on. Good note for us, Annex 1 actually states this grade A environment is normally provided by a laminar airflow workstation. Remember though, they are probably thinking of a clean bench or a clean enclosure where a wall of clean air is blowing down onto a work area or horizontally out over a process. These workstations are much less enclosed than the BSCs we are concerned with. There's grade B. We will see that it can be less clean than grade A. I usually think of it as the room that contains the grade A clean bench or BSC. Then we have grades C and D. They're even less clean and are specified for less critical stages. I think of them as support areas. The annex defines the levels of particles allowed in each grade. C grade A is allowed 3,520 particles, size 0.5 micrometers in diameter and greater per cubic meter at rest. It's allowed 20 particles, size 5 micrometers and larger per cubic meter. Remember, the nature of particles in the air is that there are always more smaller particles than larger. Notice the requirement for grade A is the same for when in operation. The limit of 3,520 particles, 0.5 micrometers and larger, is the same as the specification for ISO Class 5 in the air cleanliness standard, ISO 14644-1. ISO standard 14644-1 is part 1 of a family of standards relating to clean rooms and associated controlled environments. Some of the other parts include part 3 on test methods that describe how to measure air velocities, test filters for leaks, and more. Part 7 on separative devices would apply to clean benches. But the most popular part by far is part 1 on the classification system, which includes this table defining the different air cleanliness classes. See how for each ISO class, the limits for the different particle sizes are provided. The classes move up by orders of 10, so ISO class 6 allows 10 times more of whatever particle size it does at ISO class 5. The grades in Annex 1 are close to this, but as I mentioned before, not exactly the same in all cases. So while grade A allows 3,520 particles, 0.5 micrometers and larger, like ISO class 5, it has a limit of 20 particles, 5 micrometers and larger. ISO class 5 would have said 29 particles. See, that would be 10 times less than the allowed particles, 5 micrometers and larger in ISO class 6. But since 2015, this table does not even have a limit for ISO class 5 at 5 micrometers because the ISO people consider it too few particles to count effectively. At any rate, let's go back to the annex. So before 2015, the ISO class 5 limit for particles 5 micrometers and larger was 29. We see here grade A is 20. So technically speaking, grade A is just a little bit cleaner than ISO class 5. The limits for grade B, remember this is the room around our BSC, is actually the same as the old ISO class 5 at rest, but in operation it goes up to 100. It goes up 100 times to ISO class 7. Grade C is ISO class 7 at rest and ISO class 8 in operation. Grade D is ISO class 8 at rest and can be whatever it needs to be in operation. But air cleanliness is not really a problem for our BSCs. Let's look at what else Annex 1 says about grade A areas. Here's the rest of the description of grade A we presented earlier. Remember, this was where it said normally such conditions are provided by a laminar airflow workstation. We were feeling pretty good right around then. But it goes on to say laminar airflow systems should provide a homogeneous airspeed in a range of 0.36 to 0.54 meters per second, or about 71 to 106 feet per minute. That's a little high. In fact, I looked at the online NSF listings. I'm showing the results in a histogram to the right. The vertical axis is the number of models and the columns are for different nominal set points for downflow. The first column for models with nominal downflows between 40 and 45 feet per minute, and the second column for between 45 and 50 feet per minute each have less than 20 models. Out of 317 models of Class 2 biosafety cabinet, only one model had an approved downflow within the bands suggested by Annex 1, and that model is not currently in production. As you can see, all the other NSF-listed BSCs had nominal downflows below the guidance value, most between 55 to 65 feet per minute, 0.28 to 0.33 meters per second. And remember, we cannot go just changing inflows and downflows. A key performance feature of a Class 2 BSC is the validated balance of inflow and downflow at the front opening, which keeps contamination from the room out and hazard from within the BSC work area in. But remember, this is a should or guidance value, not a shall. We already talked about how the annex is thinking in terms of a less enclosed laminar airflow workstation. Actually, this paragraph continues on to the next page. The last sentence acknowledges that unidirectional airflow and lower velocities may be used in closed isolators and glove box. Unfortunately, they don't specifically include Class 2 BSCs, though the principle certainly applies. Okay, so the regular may be expecting 0.36 to 0.54 meters per second downflow, but it's not really required that we should expect to have to prove or validate that the downflow is sufficient. But it's not just Europe that has this view. On page 6 of the FDA's aseptic processing, we run into a similar mixed messaging on the needed air velocity. The filtered air velocity, our downflow, needs to be sufficient to sweep particles away and maintain unidirectional airflow during operations. Well, that's fine. It goes on to say the velocity parameters need to be established and justified as appropriate. More on that later, but we see some footnotes. Footnote 4 just explains what HEPA means. That's no problem. Footnote 5 moves us into that velocity issue, saying that 0.45 meters per second has been generally established. Now remember, Annex 1 suggested a downflow of 0.36 to 0.54 meters per second. Aseptic processing says 0.45 plus or minus 20%, and that actually works out to be the same. So again, we have a recommended accepted value for a grade A critical area appropriate for a clean air workstation that's a little too high for a Class 2 BSC. And again, we can point to the reason for the velocities to provide clean air and sweep contaminants away. So what else do we need to look at? Back to Annex 1. On pages 3 and 4, we see for grade A zones like our BSCs, we not only need to classify the air cleanliness, we need to monitor it. Annex 1 states particle monitoring must be done for the full duration of critical processing. A few sentences later, it states there need to be alert limits and alarms. The pharmaceutical manufacturer will probably have a particle counting system, but we're going to need to provide a sampling probe from within the BSC working area. A standard approach is a customization where a stainless steel sampling tube is placed in the rear interior wall and is as marked in red on the BSC illustration. We have also priced out a frame that would hang from an IV bar and hold a particle counter probe within the BSC work area. The user would route a particle sampling probe into the BSC through the smart port and hang it from the hanger. Remember, the smart port provides for safe, contained passage for tubes and cables through the side wall. More guidance is suggested on page 6, where it talks about isolators. Our BSCs are not isolators, but we have the same issue of not being a clean room, but acting as a grade A area. Notice there's a need to validate the technology or design. So for a BSC, we'll need to validate the quality of the air inside the cabinet, not a problem, how to clean the BSC work area, and how materials are passed into the BSC. Also, if we remember way back to the beginning of this talk, remember what makes the BSC appealing is not just the clean work area. They can get that with a clean bench or in the clean room. It's the fact there's a contained clean area. It contains hazardous drugs as well as providing a clean environment. So how do we validate that containment? How do we monitor that? Validated containment brings us back to our safety heritage. That is the focus of NSF and TÜV Nord as they validate BSCs as meeting NSF ANSI 49 or EN 12469. During the type testing for personal protection, 100 million to 800 million airborne spores are released inside the BSC and are sampled for at 8 different locations just outside the BSC front opening. No more than 15 of these spores can be recovered outside the front opening. That is a containment efficiency of 99.999985% or better. For product protection, 10,000 to 80,000 airborne spores are released just outside the BSC front opening. Notice we've completely covered the entire work surface within the BSC. Here, no more than 5 of these spores can be recovered inside the front opening. That is a barrier efficiency of 99.9995% or better. In some cases, as in NSF's case, they will do envelope testing where they will reduce the inflow and increase the downflow to assure containment even just outside the cabinet's operating range. So for GMP purposes, we know the inflow velocities at which we know we have containment. For an even better fit into GMP, many cabinets already monitor their cabinet for operation and alarms using dry or voltage-free contacts. Now, these contacts are ordinarily used to activate external exhaust systems. When the user is working with volatile toxic chemicals, they will connect the cabinet to an external exhaust system that draws the filtered BSC exhaust out of the laboratory or room and expels it outside the building. There will be an exhaust fan on the roof that pulls the air through the exhaust duct. In gang systems, they may serve multiple BSCs. The exhaust fan may run all the time, but the dampers or chokes allowing draw from this particular cabinet will only open when the cabinet is in use. Some users wanted the ability to signal the external fan to come on or the dampers to open, and that's what these remote contacts were originally used for. Like many manufacturers, we provide these voltage-free contacts on our cabinets. On our PCB, a printed circuit board within the cabinet, we have the monitor connection that will open or close, whichever you want, when the fans are operational full speed and no alarms are activated. In our case, we have a window position alarm and an airflow alarm. The airflow alarm will signal when the inflow is too high or too low, or when the downflow is too high or too low. We also have the external fan connection that will open or close, whichever you want, when the BSC fans are at operational full speed. This allows the user to have an external fan to come on whenever they turn on the cabinet and open the window. But for GMP, given the validated velocities for containment, we can use this function to send a signal when the BSC velocities go out of range. This gives our GMP customers the ability to monitor airflow for containment. I should note that different cabinets have different capabilities. Some cabinets have this capability standard, others require the purchase of additional accessories, and still others may not be able to do it at all. Returning to the GMP's required process validation. In stage 1, the user figured out their strategy, in our case determined they wanted a combination of containment and cleanliness provided by a Class 2 BSC for their process. In stage 2, the user designs the process, identifies the equipment and needed performance. In this case, they will specify a Class 2 biosafety cabinet with the ISO Class 5 Grade A work area. They will specify validated airflows as provided with NSF or TÜV listed Class 2 BSCs. They will sample the plan for monitoring. They will specify the plan for monitoring performance, including air cleanliness monitoring through an air sampling tube and containment through use of the voltage-free contacts. When the BSC arrives, they will need to validate or prove these things work, that the airflow operates at ISO Class 5 Grade A as they do their process, and that the process can be monitored. And then when they move into production, they will need to monitor these things, use the cabinet, clean the work area as needed, and so on. Class 2 biosafety cabinets are a valuable tool when cleanliness and containment are required in a GMP setting. But like most tools, we will need to do some work to get the most benefit. First, make sure the BSC has validated performance. For example, in Europe and North America, there are recognized national laboratories that carefully review the BSC design, validate the cabinet's design's ability to contain hazardous aerosol, and provide a clean work area, and continue to monitor participating cabinet manufacturers with factory inspections and so on. We do not know the cabinet has the ability to do what we need just because someone somewhere calls it a Class 2 BSC or even claims that it meets one of the international standards. We need that independent verification or validation of that. Second, we need to bring that design into our GMP environment. We did not have the opportunity to talk about this, but both EN 12469 and NSF ANSI 49 recommend a field certification of cabinet performance, especially at the first installation. We'll need more than that, but the performance we need starts with that. The service engineer will properly measure and set inflow and downflow velocities, check filters for leaks, verify airflows within the room do not compromise the cabinet's air barrier at the front opening. In addition, these service engineers will help set up the airflow monitoring. For instance, in our case, they would set the inflow alarm to the value at which containment is validated. They would set the downflow alarm to standard parameters. Once we have the alarm set properly, we have the opportunity to use the potential free contacts. Next, as I read Annex 1 and into the FDA's aseptic processing, it seems like we're going to need to do particle monitoring within the BSC work area, and that means a sampling tube. Finally, as with all equipment in the GMP environment, we will need protocols for how we will clean and use the cabinet in our processes. Well, thanks for your time. I hope you found it helpful, and now I'm ready for any questions or comments you might have on how BSCs can be used or effectively used within the GMP environment.
Thank you, Dave. Thank you for the talk. So now I think we are able to open the time to the floor. So please feel free to type in your questions in the Q&A box. For the time being, I have received a few questions, so I would start probably first with this one. So I think it's about the particle counter. So so you mentioned on one of the slides, well, when we are referring to the ISO 146 44, the table refers to different air cleanliness standard based on particle. So 0.1 micron, 0.2, 0.3, 0.5, 1 micron, and even 5 micron. So when people are selecting the particle counter, what type of a counter should they get, and what particle size should they actually monitor?
Yeah, well, it's pretty clear in GMP that they really like the 0.5 micrometer size and the 5 micrometer size. Although when you class something as ISO class 5, you can measure, a difference. You can measure 0.1, 0.2, 0.3, 1 micrometer. But in Annex 1, they specifically say you've got to measure 0.5 and 5. So that's the first point. You would definitely want to get a particle counter that can measure those two sizes. Second, I can't remember the name of the standard, but there is a kind of a recognized standard for particle counters these days. I'll have to get that information to you later, Jeffrey, but it's an ISO, and then there's a bunch of numbers that come after it. And what that means is that particle counter kind of meets the international standard for what particle counters should do. And you definitely would want that, but you wouldn't want to have a counter that didn't do that, because it would certainly raise questions about your data.
Okay.
Oh,
So
and then sampling, you would need to sample at, I think, 28 liters per minute. But yeah, that would be all.
Okay, so I think the follow-up questions on the particle counter. So do we have any specific requirement about the location where the particle counter should be located inside the cabinet?
Sure. The guidance in aseptic processing in Annex 1 has good news in the sense that they recognize you can't put the probe right in the middle of what you're doing because it would be disruptive and would pick up stuff from that. But they do want to measure air very, very close to where your work is. With a biosafety cabinet, it's pretty easy. It would be about the height of the top of the window, maybe between 0 and 1 centimeters above the top of the window opening, and in the center, which would be where your work is. If you were working to one side, you could move it over there. But in general, just above the work area, above where your hands will be, where you'll be working with samples.
So the general guidance is probably 0.1 meter above the window opening and is located at the center of the cabinet, right?
Yes, yes.
Mm. Okay. So another question says, is there any signal to tell operator when a filter needs to be changed?
Um, well, one, it's going to be, you'll probably retire from the company before your cabinet will need a filter change. Because remember, you're in a grade B area, most likely. And in a grade B area, the air is pretty clean. HEPA filters and biosafety cabinets normally last, you know, well over 10 to 15 years. So point one, it's probably not going to happen very quickly. Point two, what's usually required in GMP environments is that the cabinet is certified semi-annually every 6 months. And so when the certifier comes in and measures the air velocity and sets the fan speed, they will be able to tell you if the filters are going to need replacement. So beyond that, if it affects the airflows, cabinets with airflow alarms like ours would signal you if that was a problem. But again, it's very unlikely that those alarms would ever go off. You would get advance notice from the field certifier in those relatively infrequent times where you would need new filters.
So so the signal is actually coming from the semi-annual certification, right? So the engineer will tell you whether the filter needs to be
Yes,
changed.
yes,
Okay.
that will be the most common situation.
All right. Um, let's see. We have a few more questions. Um, so this one is about um, the monitoring. So the question is, unlike a CO2 incubator or a freezer, biosafety cabinet is very much an attended equipment. So that means the equipment is run in the presence of an operator. So why it is still required to monitor the operation of the cabinet? Is it for auditing purposes, or is it part of the process validation requirement?
Well, and that's a good question. And for many years, my position was, it wasn't necessary to monitor it externally. But when you read aseptic processing, when you read Annex 1, I mean, they really want to monitor it. They really want to monitor it. And if we talked about, if you, as I was explaining, um, it's going to take, as you prepare to use a BSC in a GMP environment, you may need to explain why you're using it, what it's doing, how you know it works, and that those explanations will go a lot better if you say, in addition to the audible alarm, which the user will listen to and react to right away. There is you're monitoring it electronically, and then you'll have access to that data also. So normally I would agree, but GMP is such a documentation intensive process. I think we need to go one step beyond, which is part of that translating the BSC from the safety universe into the GMP universe.
Do you see actually this is part of the auditing requirement? So when the auditor came in, they will look at these performance data even
Yes.
for a carbonate?
Yes, and see, they will be happy when they see it. So if I'm the auditor and I come in, I go, okay, I see what you're doing. Okay, how do you monitor the airflow? And I say, oh, it's easy. It just alarms when there's a problem and the users and the operator stops working. And as the auditor, I'm looking at you and say, so do you have any records of that? do you and I'm a little uncomfortable. I mean, you might be able to persuade me, but I will be really happy if you can then pull out this nice sheet that say exactly, whenever the cabinet stops performing. That's the kind of information I expect to see. So that's kind of why we're trying to do that.
Okay, all right. So here's another question. So I think it's all about the particle counting as well as the downflow value. So you said that is it for the aseptic process, the downflow value in the carbonate should be between 0.36 to 0.54 meter per second.
Well, yeah, see,
So
that's our problem. Is
mm,
that the minute so let's say we take a cabinet, it's not designed to work at those high speeds, because I just showed that almost no cabinets are designed to work at those high speeds. And then we just turn up the speed. We just increase the downflow. Well, now that validated containment at the front opening that NSF or TÜV Nord or whoever proved at the design velocities, that's not relevant anymore. And so we can't, in a sense, prove the containment that we wanted it for. Now, I've seen some customers basically say, I don't want to explain why my velocity is lower than that, so I'm just going to run my cabinet wrong. And they do. I disagree. As you hopefully picked up in the presentation, the guidance documents in Annex 1 and in the FDA's document, aseptic processing, they open the door for more contained work areas to have lower velocities. And that would be part of that important documentation step at the beginning of the validation processing that I would recommend.
Okay, so actually the question is a combination of both the particle counting and the increased downflow. The question is, if the particle counter already shows the air inside the carbonate already meeting the grade A cleanliness,
Sure.
do we still require to increase the downflow to 0.36 to 0.54 meter per second? Are these two conditions they need to coexist at the same time, or either one of them is already good enough for
Well, it's
GMP?
not right. It's not either or. I mean, the air cleanliness is the absolute requirement. So the grade A we need. The air velocity is a guidance value. They don't say it now. Some regulators don't notice that it's a guidance value. You notice in aseptic processing, the velocity just needs to be fast enough to sweep the particles away. And they said usually that's 0.45 plus or minus 20%. So no, you definitely have to do the particle counts, the air cleanliness, grade A, but you do not need the velocity. But my recommendation would be to write your explanation about why that's okay. Don't assume that nobody's going to ask you about it, because it's quite possible that they will ask you, well, gee, I notice your velocity is only 0.32. Why don't you increase it? And you would need to be able to say, oh, because uh, we want the validation of containment, which was at this, and then you go into that whole thing. And for I'm sure any manufacturer would do it, but for people who purchase our cabinets, we would help them with that language.
Okay, so here is a question from I think probably from an existing user. So they have got one of our cabinet. That's the one which you show on the slide, the 2030. So he's asking how they're going to do this operation monitoring. How can they do that?
Yeah, well, if they read in the, I forget whether it's in the operator's manual, service manual, how to signal an external fan, you will find the instructions that I showed on the screen, that explain when these potential free dry contacts open and close. So what we want to have a signal for when the fan is on and there are no alarms. And by using the two remote contacts that I talked about, you can come up with a circuit that will basically be open when that is true. And when that is true, you can do your work. And when the fans aren't on, or when the fans are on and there's an airflow alarm or a window position alarm, you can't do the work. And that's what I mean by monitoring. And I can send an email to them if they would like more guidance on that.
Okay, all right. So here's another questions. Is there any requirement on the microbial cleanliness for GMP? Is there any common practice you can suggest to validate the microbial cleanliness?
Well, okay, there's two. I don't know as much about this, but there's two issues. There's airborne microbial load, which you can sample for that. There doesn't appear to be, in the reading about monitoring, the sense I get is the regulators were saying, hey, we know monitoring microbial cleanliness doesn't make much sense. If you monitor for particles, that will tell you microbial. So that's one. Now, in addition to that, or separate from that, part of your validation process will be some wipe or swab testing or things like that to prove that your the cleaning regime you have removes any microbial contamination. So I would think those would be the two ways. Periodically you might sample inside, but you're never going to find anything because there's hardly any particles. And then you're going to have to address the degree to which you clean or demonstrate your effectiveness in cleaning the work surfaces.
Okay. So we have another question occasionally. It's coincidentally, it's also about cleanliness. So do you see any need to give a BSC a stainless steel exterior in order to meet certain air cleanliness or cleanability requirement in a GMP setting?
No, I don't. However, I know the regulators like seeing stainless steel. It just, when they look at it, they go, oh, that looks familiar, and I'm happy. In reality, for what we're doing, the work is being done inside the cabinet. The cabinet interior is either all stainless steel or stainless steel and with some glass. So you are going to need to address whatever your protocols have for cleaning like the room, you know, how you clean the walls and things like that, I imagine those same requirements would carry over to the exterior of the biosafety cabinet. But just as your walls don't have to be stainless steel, I don't think the exterior of the cabinet has to be stainless steel. Though I recognize it would probably make the regulator smile when he saw that.
All right. So I guess this is probably the last question. So you mentioned on your early slides that a biosafety cabinet can easily reach ISO class 5 or class 4 or even higher. Do you see well we occasionally see certain supplier, they say they could guarantee a cabinet can meet ISO class 3 air cleanliness. What would you think about this statement? Is it a valid statement, or it still has to be validated by a particle counter?
Well, there's two questions there. The first question is, will a regulator be happier with ISO class 3 or 4 than they are with ISO class 5 grade A? And there's no indication of that at all. The regulations say we want grade A. You know, there's no such thing as a grade A plus. You know, the regulator feels like if you hit ISO class 5 grade A, you're fine. So there's no real benefit to going beyond that. So that's first part. When it comes to GMP, now if we were making, you know, computer chips or microelectronics or things like that, then those people start to really want the cleaner air cleanliness classes. So that's the first point. Second point, I think almost all you know, properly performing class 2 biosafety cabinets, you know, that have been validated by NSF or TÜV Nord or somebody like that, hit ISO class 4, ISO class 3. So I could see a company claiming that, because I don't see that as a particularly unique thing to claim. The reason why you don't see it more, it's just a royal pain to do the sampling. You have to sample. I actually did it in our research center just for fun, and I had to I think I had to take like two 35-minute samples to sample enough air to be able to and I think I was just sampling 0.3 micrometers inside, you know, to be able to document that it hit ISO class 3. And nobody wants to spend that time. It only takes like a minute to sample the air for ISO class 5. So point one, you don't need ISO class 3 or 4 in a GMP setting. Point two, almost all biosafety cabinets do it, so it's not really worth bragging about, in my opinion.
Thank you, Dave. I think that's we are approaching the 11 a.m. It's almost close to the end of the hour. So I think thank you, Dave, for your time, and I hope all the audience you enjoyed the talk a lot. Before the close of the call, allow me to give you a one more notice, and that is the third event is coming, and the third live webinar will be on next Thursday, November 5th. And the speaker will be Josh Lewis, one of our senior products for remote monitoring. So his talk will be on managing data in laboratory equipment. It's talking about connectivity solutions and their regulatory implications and quality control. So thank you everyone for coming to the talk. So I hope you enjoyed, and hope to see you all next week. All right. Thank you. Thanks a lot. And thank you, Dave.
Thank you, everyone.
Okay.
Sure thing. Bye-bye.
Bye-bye.